I'm Dr. Joshua Kohn. Up until last June, I was a professor at UCSD in the Department of Ecology, Behavior, and Evolution. My field is plant sexual systems, but for the last 5 years or so, I've studied feral honeybees here in San Diego County, and that's what I'll be talking about tonight.
Good evening, everybody. I am Cheryl Goddard. I am the Executive Director for the San Diego River Valley Conservancy, and I welcome you here tonight. It is our speaker series connecting creatures, climate, and communities, dedicated to exploring the intricate connections we share with our ecosystem, including the landscape, plants, land and marine animals, insects, and geology. Tonight, we have the honor of hearing from Dr. Josh Kohn on the good, the bad, and the complexities of non-native honeybees. And tonight is the last talk of our 5-part speaker series. I would like to thank our previous speakers. We have Chris Corey here tonight. He provided us his perspective of working with the Conservancy and the San Diego River Park JPA over the last 4 decades. We had Dr. Drew Talley, who enlightened us about the killifish and how they are indicators on the health of an aquatic ecosystem. We had Serge Dedina, Executive Director from our wonderful partners Wild Coast, who shared their work on their blue carbon programs down in Baja California, up the coast of San Diego. Last month, we had Mike Thacker over at the Santa Isabel Nature Center, who shared his knowledge about early geology formations of San Diego and also led us on a hike pointing out his favorite formations around Santa Isabel. It's been wonderful hearing all the speakers who are gracious enough to share their knowledge and perspective with our community. I'd also like to express my gratitude to Karen Searcy, to Kim Yinks, who was checking everybody in, and to Gabrielle Weinhausen, who couldn't make it here tonight. She's out visiting family in Germany. But their dedication took this brainstorming idea of a speaker series from just discussions to reality, and we had a 5-part wonderful speaker series because of them. And they are busy working on our 2025 series, which will begin next February. I'm also pleased to share that we've recorded all our presentations, and they will soon be available on our YouTube channel for easy viewing, sharing, and accessibility for our community members. And before Dr. Kohn takes our stage, I would like Karen Searcy to come up to give him the well-deserved introduction. Thank you.
Well, it's my great pleasure to introduce Dr. Josh Kohn. He grew up in Berkeley, California, which is very dear to my heart because I was born there. I think one of the more interesting things is that while he was still in high school, and for the years thereafter, he worked with Chez Parnasse with Jeremiah Power and Alice
Waters. I figured some people in the audience would know those people.
Oh yeah, it's impressive.
It was a joyful time.
Read rather than okay, so he graduated from Reed College in Portland, Oregon, where he received a bachelor's degree, and then worked as a field biologist on birds projects in the Sierras and in Hawaii, and decided that plants were easier to look at than birds.
You can find them again.
They stay still. So he earned a PhD at the University of Pennsylvania, and then has spent the last 32 years as a professor of biology at UCSD in the Department of Ecology, Behavior, and Evolution, and he retired just last July. So we're very lucky to have him this evening, and he's going to talk about the effects of superabundant non-native honeybees on plants and pollinators.
Thank you, Karen. So I'm honored to talk to SDRVC because I consider them among my best neighbors, if not my best neighbor. I first learned about the Conservancy when I was working on a DNA barcoding effort on insects. So DNA barcoding is a way of sequencing just a just a short amount of DNA that will uniquely identify species. So even for tiny little insects that nobody knows what they are, you can tell whether it's the same or different from the next one by how divergent they are at DNA. And I set up insect traps at 5 sites within SDRVC that span the range from the lagoon over there to the lower slopes of Kohn Mountain, and that gave me a really great introduction to the scope and the vision that SDRVC represents. And the second way that I consider them a good neighbor is that, like many of you during the lockdown, I spend a lot of time on walks that I could do from my house. And one of those is Gonzales Canyon, or also known as the Torrey Pines Loop Trail, which SDRVC runs. That starts at the dog park behind Torrey Pines High. If you haven't used it, please do. And I, in fact, just a couple of weeks ago at the bottom of Crest Canyon, ran into the SDRVC ranger who fixed the bridge and reopened the trail, which had been closed for many months prior. And so on both counts, I consider SDRVC a wonderful organization with a huge vision, and really important to try and control a whole watershed under the difficult conditions that we have here in Southern California. All right, I hope everybody can hear me fine, because I really don't like microphones. And I urge you to ask questions at any time. And there'll certainly be plenty of time when it's over. So I'm going to begin with a kind of crotchety slide that this guy, Jerry Kohn, who's a famous evolutionary biologist, came up with to describe people's careers. I only got to the red line there, which is talk about my students' work. And this is really a ruse to say that most of the work you're going to see today, a lot of it was done by Dylan Travis, my last graduate student. Some of it was done, the genetics of honeybees was done by Danny Zarate. She was my last student until I met Dylan, and then he became my last student. And a former student of my colleague, Dave Hallway, James Hung, who did really groundbreaking surveys of the bee fauna of San Diego County some years back, who I published with quite a bit. And I would say that this is all tongue-in-cheek, except when I arrived at UCSD, as you all know, across the street is the Salk Institute, and on the faculty there then was Francis Crick, the better half of the double helix. And he, late in life, had turned into something of a neuroscientist and was indeed studying consciousness. So I can't hold a candle to the discoverer of DNA, and I never progressed that far, but I really want to say that most of the work I'm going to talk to you about, at least the hard actual gathering of the data, was done by these students. Okay, this is going to look kind of scary and weird, and I urge you not to worry about it, except there will be a quiz after. It's to introduce you to the kind of biologist I am, which is not a bee biologist, although now I have somewhat of a claim to being a bee biologist. I studied plant sexual systems. The first one, the top line there, is a sexual system called gynodioece, where there's 2 sexes in the population, but those sexes are females and hermaphrodites. And the plant I worked on then was Cucurbita fetidissima, which is native here. Some plants make both male and female flowers, and other plants make only female flowers. And you can describe the sex ratio with an equation, and there's some data there that I gathered as a PhD student that explained why you'd want to give up male function and become a pure female. The second line there has to do with heterostyle. This is in the system there, there's 3 distinct types of individuals. They're all hermaphrodites, but they have different arrangements of anthers and stigmas. Darwin, that book there, Darwin wrote a whole book about this, except one chapter was on gynodioece and the rest was on heterostyle. I worked on the dissolution of heterostyle, the evolution of selfing from an outcrossing mechanism. And then much of my time at UCSD was spent working on what's called self-incompatibility. About half of all plants in the world can recognize and reject their own pollen in favor of mating with other individuals of the same species, but not themselves. And there's a bunch of theory behind that, and the stuff we mostly worked on came to the conclusion that if you evolve away from this and become a selfer, that's a sort of evolutionary dead end. You can see that none of this has a lot to do with bees, except the cucurbita up there is actually pollinated by something called a squash bee. I may get back to that. A specialist on that genus. But instead, I did science one of the ways you can do science, which is find an interesting problem. These are all problems that were written about by titans in the field, and then find the system to study it that's appropriate, and then test some theory, because I'm primarily an empiricist and not a theoretician. And that's a fine way to do science. There's nothing wrong with it, but many of my mentors early in life as a biologist were really strong natural historians. And the ones that were professional scientists often found the things they wanted to study by making an observation in the field and then going further at it given their training in the field of evolution and ecology. And I always felt a little inadequate because I never worked that way. I would go outside, have a great time hiking. Would I come back with a question for research? Never. Until it finally happened to me. And it happened in the following way. For one reason or another, having to do with family, I had to spend a couple of days in Orange County waiting for my daughter to finish lifesaver training in a pool. And so what do I do all day? Well, I can't come back to San Diego. It's too far. So I went hiking in the mountains to the east of San Clemente. And what I noticed was it didn't matter how far out into the wilderness, and there's some pretty wildernessy areas out there, how far from agriculture, how far from human habitation you got. If something was blooming profusely, be it ceanothus or any of the other native species, it was dripping with honeybees. Just dripping. And so my kids were in high school in those days, and they had a certain way of quantifying things, and this is it. And this immediately set me off a little bit because actually that James Hung, the last person I pictured there, was already working on native bees around San Diego County. And I knew from him that there were a lot of them. In fact, there's over 600 species of native bees in San Diego County. Do you see them when you're looking particularly at abundantly blooming things? Almost never. And I'll give you the data on that in a minute. So that was one worry. And the other worry is, yeah, these are non-native honeybees are a perfectly fine livestock, but they've rarely been looked at as an invasive species. And what are their effects then on both plants and animals here? And that's what I'm going to talk about today. And it's on the background that we have an extremely diverse flora. Something like 2,400 plant taxa just in San Diego County, which is why I can't name 95% of them. And sometimes that number, 650, is raised to 700. I don't know where we are now, but there's a lot of bee species also. This is a map of what is called hotspots of endangered biodiversity from around the globe, and you can see that Southern California shows up. If I used to do this in all my teaching at UCSD. At the end of the semester, I'd have everybody repeat after me that San Diego is a hotspot of endangered biodiversity. So public understanding of this is increasing, if slowly. So we got a lot of honeybees. I was convinced right from the start, and I'll show some genetic data on this, that nearly all the honeybees that you see in San Diego are feral, not from managed hives. The first question, I went back, I talked to my colleague who studies social insect invasions. This is Dave Holloway. And he said, well, isn't everywhere like this? And I said, I don't think so, but who knows? And so the first question is, if you go around the world, how many honeybees do you see on native vegetation in different places? And we'll answer that one in a second. Then we want to know the genetic makeup of these honeybees. Many of you may have heard that something that's sometimes called the Africanized honeybee has invaded this area. I'm going to call it Scutellata hybrids because there's a lot of social connotations to calling it African and then thinking about it as killer bees and aggressive and all this stuff, and we'll just leave it to the scientific term. So I'll explain what a Scutellata hybrid is. And it would be interesting if we could answer the next question, which is why are they so successful here? So any of you raise bees? Okay, that's good because I might offend you. But it's bad in that if you do raise bees, you know that when you raise bees, you often have to supplement them with extra sugar for the wintertime, and you're often treating them for various diseases. These feral bees get none of that, and yet they're super successful. Okay, and in fact, my last grant before I retired was on honeybee health, because it's possible that these bees that we have here can get past many of the diseases and other things that are afflicting honeybees that are managed and might actually be interesting breeding stock. So I'll touch on that a little bit. I can't answer this question about why they're so successful, but I will show you that they are unusually successful here in San Diego relative to nearly everywhere in the world. And then I'm going to get into how do they affect native plants and how might they be affecting native bees. Now, somewhere in here, I need to stop and tell you about native bees, so I might as well be now. The bees that you're used to are typically when somebody says bee, they mean honeybee, and that's fine, but it's, in some ways the most unusual bee there is. Highly social. Unlike almost every other type of bee, it can communicate both the direction and the quality of a resource to other nestmates because it's got that waggle dance that von Frisch figured out. The typical bee species is not social. It's what we call solitary, and that means that each female bee provisions a solitary nest. There's no worker caste. Each female bee is an independent owner-operator of her reproduction, and so goes out and collects pollen and nectar, forms it into a ball, and most of them nest actually in the ground, where they dig a hole with chambers and they put a glob of pollen and nectar in each chamber for their larvae to feed on. So most are solitary, some are social, like the bumblebees that we do have, but the vast majority of that 700 species or 650 species are solitary bees. All right, first question. Do we have more honeybees here than most other places? So fortunately, there was a database on what are called pollination networks, from which we could draw. The pollination network means you go out to each plant in a natural community and say what's visiting it at what frequency. So you get all of the relationships between plants and their various pollinators. And mostly that's used to figure out things like, well, if 25% of the pollinators go extinct, will the plants follow, or do they have enough backup mechanisms with other pollinators that they'll be okay? But we used it just to ask what's the frequency of visitation by honeybees that we see here relative to other places in the world. So here it turns out across all plants, 84% of visits to flowers are by honeybees. And that's on this slide, that's where the blue arrow is, that's us, and the green part of those pie charts is honeybees. And you can see there's very few pies that are as green as that. There's a couple on oceanic islands, and there's one site in Kenya. Otherwise, we're leading the world. These are all data taken from natural habitats in different, 80-some studies in different parts of the world. So the answer is, yeah, honeybees are unusually successful here. And in addition to that, the more abundantly a species is blooming, the greater the difference, or the greater the percentage of visitation by honeybees. So these two, I wonder if I have a pointer here. Nope. These two regressions are flower abundance on the x-axis. These are similar data taken in slightly different ways but show the same thing, and observed number of visits on the y-axis. And the filled circles are honeybees, and the open circles are native insects. So you can see that when a flower, a species is not blooming abundantly, you get kind of equitable visitation between honeybees and the other things that are around. But the more a thing is blooming, think of a big patch of whatever you can think of, the more it's dominated by honeybees. Those solitary bees hardly increase at all between sort of rare plants or infrequently blooming plants and abundantly blooming plants. But honeybees can tell their their sisters, hey, out in this direction there's a great resource, and they'll just dominate the heck out of it. All right, so what are these bees? So this graphic here shows you the invasion of what's called killer bees, but I'm going to call Scutellata hybrids. In the 1950s, a variety of honeybee, a subspecies of honeybee called Apis mellifera. Apis mellifera is the Latin binomial for honeybees. Apis mellifera scutellata is a honeybee that comes from southern and eastern Africa. And it was brought to Brazil by a bee breeder in the hopes of breeding a bee that would be more adapted to tropical conditions. And unfortunately, they did both hybridize with local bees and escape. And that hybrid thing that's called Scutellata hybrids has from 1956 been migrating both north and south. To the south, it stopped somewhere in Argentina, but to the north, it's been going decade by decade or year by year quite rapidly until it hit Texas in 1990 and San Diego County in 1994, and now extends. It gets lower in frequency, but it extends as far north as right around Sacramento. And what else is on this slide is on the left, you see representatives from the other three major lineages of honeybees. So honeybees are split into subspecies. There you see three of them on the left, and then that African one, Scutellata, on the right. So these are these that were identified early on by both morphology and genetics as sort of distinctions within the species Apis mellifera. And all of the honeybees in North America prior to 1956 were made up of those three groups that had been brought. Yes?
If you're going to get to this, great, but I'm just wondering if you can explain how the bees that are brought in to pollinate agriculture in California fit into it.
Yeah, great question. Let's hold it for the end on that one, because I'll have quite a bit to say, but I need to get through some of this first in order for that to make sense. But yes, for commercial hives or commercial beekeeping, by and large, it's not for honey. It's for pollination services. And it's particularly for things like almonds and apples. Both of those, by the way, are self-incompatible, so you have to move pollen between trees to affect fruit set. And because they're very densely grown and they only bloom for a short period of time, you need to bring pollinators in, and they bring them in by the 18-wheeler load from all over North America. And most of the bees managed, the bees that are managed in North America are managed for that purpose. And we'll get to what problems they may or may not be facing at the end. All right, so we have the invasion of these Scutellata hybrids. What was resident was some mixture of the other three. Those were brought well in the 1500s to the East Coast, and just after the gold rush to the West Coast. There's a very interesting San Diego connection to that. Ask me at the end. All right. So these days, people do genomics. This is the only genomics I've ever had done in my lab. It was very painful because I couldn't really even advise the student on how to do it, but she got other people to help her. And it's called an admixture plot. It's Each column there is an individual bee. The first set that look all red, those are all individual bees from Africa. They represent that Apis mellifera scutellata, and we're going to put them in red. Black there is these are bees from Western Europe. They're mostly one thing, and they're considered those black. The yellow there is from Eastern Europe, and the aqua there, those are bees that are from the Middle East and Anatolia. What about bees in the New World? So my student Danny collected bees from Panama, Costa Rica, Mexico, and San Diego, and then we can ask what fraction of their genomes come from Africa versus the various European and Middle Eastern forms. And what you see for the three to the farthest right, the ones from Central America and Mexico, most of their genomes come from Africa. That's why they're mostly red. The black there is Western Europe, and the yellow is Eastern Europe. So they're roughly 80% African. But you get to San Diego, you go collect bees, and everyone you collect has roughly a little less than 40% African, and the rest of the genome is made up as a mixture of all of these three other forms that were introduced previously. So they're incredibly mixed up genomes and quite distinct from the genomes of anything else. And this could have something to do with their success. We don't really know. This is just another way of looking at it. These are various measures of what's called genetic diversity. How much variation is there in the genome? And here what's interesting is the African form on the left has the highest level of genetic diversity. The various European and Middle Eastern forms have much lower levels of genetic diversity. The New World bees, most of which have 80% African, have high genetic diversity just like Africa, but San Diego bees also have high genetic diversity, not entirely because they have African parts to their genome, but because they're a mixture of all of these forms that came from different places. So again, these are different measures of genetic diversity. They all say the same thing. And our bees are unique in representing all four of these major lineages. And it's possible that this high level of genetic diversity, as well as the fact that the genes they have come from different parts of the world and may have been selected in different places for different things, that may be one reason that they're as successful as they are and as able to be successful in the face of the diseases that plague managed honeybees as they are. All right, now we're going to get on to the meat here. We've got these unusual bees. They're super abundant. How do they affect plants? So for this, we worked on 3 native species. The first 2 are black and white sage. You might notice that their Latin binomials are Salvia apiana and Salvia mellifera. And honeybee is Apis mellifera, so their connection to honeybees was recognized as far back as when they were named, which was in the early 1900s. And then Phacelia distans, which is it's common where it occurs, but it's not as widespread. Black and white sage can be major pollen-producing plants on the habitat. So one thing I failed to mention when I was describing solitary bees and bees in general is that all bees are made of pollen and nectar. At least there's some exceptions in the tropics where we have bees that eat carrion, but every bee here is made of pollen and nectar, so they all share that in common. That'll become important for the second part of the talk. Anyway, if you go out now, Salvia mellifera, black sage, sort of starting to finish up. White sage will be coming along, and Phacelia is probably right in the middle of blooming where you see it, but it's a bit less common. These are all things that locally can be super abundant, can represent most of the flowers in a given place at a given time in the season. So they're things that honeybees can gang up on, and in fact they do. So this shows you the proportion of visits or visitors from honeybees in whatever that color is, pinkish, and from native insects in blue. And so for Salvia apiana, 93% of all visitors are honeybees. For Salvia mellifera, it's up to 95%. And for Phacelia distans, which is a little less common, it's at about 80%. So these numbers in the 90s, that's what you'd get in an orchard of almonds when you brought honeybees in for the purpose of pollination. Here we have similar levels of numerical dominance by honeybees. It's not due to managed hives, it's due to feral hives. So it's a stunningly abundant honeybees here. One thing I noticed right off is that native insects tend to be flightier. Everybody loves to watch honeybees visit flowers because they're slow as molasses, and they go flower to flower. They're super methodical. And so they visit a lot of flowers on a plant before they move to the next plant. And in fact, they visit roughly twice as many flowers on a plant before moving as does the average native insect. So why would I care? So here's a word you can use in the bar the next time you go out. Geitonogamy. Only Karen knows this word. Geitonogamy means moving pollen from flower to flower on the same plant. It's the chief form of self-pollination among plants. That's the way most self-pollen ends up on a stigma, is through movements between flowers rather than movements within a flower by the pollinator. And that can change the rate at which the plant self-fertilizes. So here there are some data, not from me, but from a colleague in the Midwest who followed the sequence of visits to a certain plant. It was a Mimulus ringens in this case, and measured the rate of self-fertilization, which means what proportion of all seeds were due to selfing versus cross-pollination, for the first flower visited, the second, the third, and the fourth. And you can see that the selfing rate varied from 0.2 to 0.7. So that means on the first visit, 80% of the seeds were outcrossed. And on the fourth visit, only 20% or so of the seeds were outcrossed. Again, why do I care? Well, what happened to the royal families of Europe when they started mating with their cousins?
Yes.
Anybody? Bad things happened, okay? And those inbred offspring were less fit than the outbred offspring. So fitness, when I say it, means how many seeds do you set? How many of those germinate? How well do they survive? How many offspring do they produce? How many genes are passed to the next generation? That's what we call fitness. And many plants have been measured for their level of what's called inbreeding depression, the difference in the fitness between self and cross-fertilized offspring. And often that difference is well in excess of twofold. And the reason it's been measured a lot is that field I come from, plant mating system evolution. It's one of the strongest selective forces for, say, outcrossing or selfing, depending upon the level of inbreeding depression. If inbreeding depression is high and if selfed offspring are lousy, then you should outcross. And if selfed offspring are just as fit as crossed offspring, you should self-fertilize. So it's been studied a lot, and you often get whacking great differences in fitness. So now I'm starting to think, maybe honeybees, because they're so methodical, so as they move from flower to flower, are causing more selfing and causing lower fitness of offspring, and that's where we're headed. All right, so we measure all these things. Wait, we, that's the royal we. I just think of things to do, and to my great delight, Dylan went out and did them all, and they're super tedious. And this is sort of field ecology in a nutshell. I mean, it sounds glamorous when your laboratory is outside, but really it's a lot of tedious work. So he measured how much pollen is deposited on a stigma by a visit from a honeybee versus a native insect. How many seeds are produced from a visit? How well do those seeds germinate? how well do they grow and survive? This part we're doing in a greenhouse. And for the annual species, that Phacelia species, how many flowers do they produce? So for the perennials, we use growth as a measure of fitness because size usually translates bigger size, more flowers. But because it's a perennial, we couldn't grow it forever. And for the annual, we could actually get a measure of how many flowers they made. We also made Dylan made hand, self, and cross-pollinations to follow the effects of that and compare it to what you get between native insects and honeybees. So here's all the treatments. Those shaded areas around the inflorescences, those represent mesh bags that prevent pollinators from visiting. So you bag the inflorescence, and then when the flower opens, you take the bag off and apply one of these, I think, 6 treatments. Either you hand self-pollinate or you hand cross-pollinate, then you put the bag back on, come back later, get those fruits, use those seeds. Or you allow a single visit from a native pollinator or a native insect, or a single visit from a honeybee. We also had, do they need pollinators at all? You could just leave the bag on. They need pollinators. They set very few seeds if you don't have a pollinator. And we had an open pollination treatment, which is just don't use a bag at all and let them get the pollen that they get. That's hard. Okay, then you go and you measure seed set per flower, and then you start in the greenhouse measuring germination rate, the fraction that survive. In this case, we went to 10 weeks. The leaf number as a measure of size at 10 weeks. And for the annual species, which is the Phacelia, the total number of flowers they produced in their 1 year of life. And our measure of fitness is just multiply all those things together. So this is standard practice in evolutionary ecology. You gotta be a seed that sets. That means that after fertilization, the seed develops. You gotta be a seed that germinates, survives, grows, reproduces. So, as far as pollen deposition goes, there's no statistical difference between native pollinators and honeybees, although native pollinators seem to give a little bit more pollen. Both types of pollinators give plenty enough pollen in a single visit to achieve full seed set. Full seed set in any of these species is just 4 seeds per flower. And you'll notice that we don't have a measure in black sage, Salvia mellifera, for native insects, and that's because honeybees were so dominant, we couldn't measure what the effects of a visit by a native insect was. So for 2 of these species, we have all the treatments. For 1 of these species, we lack the native insect treatment. All right, so this is the big take home, but it's a little too complicated to go through, and I'll take it step by step. These plots are called box and whisker plots now. The heavy black dot is the mean value of, and the bar is the median value. So you can just sort of look at those. And in any of these, if you're comparing treatments, just look at the letters. If the letters are the same, there's no statistical difference. And if the letters are different, then there's a difference between the treatments. And I'm going to go sort of comparison by comparison because even I can't take this all in at one go. So first, what about self versus cross-pollination? How much inbreeding depression is there? So in all 3 species, you'll see that the cross treatment, hand cross fertilization, has the highest fitness and much higher than hand self fertilization, which is the next bar there. And in fact, cross-fertilized offspring are 2 to 10 times as fit as self-fertilized offspring in all 3 species. So these are whacking great levels of inbreeding depression, but not surprising. You see this in plenty of plants that have been measured. What about cross-fertilization versus native insects? Again, we could only do this for 2 of the species, and this time you'll see that they don't differ. Native insects, pollination leaves a seed that is as fit as a cross-fertilized seed. Now we're highlighting self and honeybee, apis, and open pollination. Remember, open pollination, especially for the 2 salvias, is almost entirely honeybee pollination. So self-pollination gives you low fitness and not different from a single visit from an honeybee in any of these comparisons. And then the open pollination treatment, just let insects come at willy-nilly, for the 2 salvias is not different from self-fertilization. For the Phacelia, which gets a fair bit of visitation from native insects, it's a little bit higher. But the real thing that we were hoping for and going after, what happened there? What's the comparison between being visited 1 time by a honeybee and 1 time by a native insect? And there we get, in the 2 species that we could do the comparison, native insects visitation gave you fitness values for the seeds that were 2 to 5 times as high as visitation by honeybees, even though, as we've seen, honeybees are doing the great majority of all of the pollinating of our plants. All right, so for this first part on the plants, yeah, the take-home message is honeybees are bad for plants. Who knew? They visit more flowers per plant before moving on than do native insects, likely increasing the selfing rate. And the fitness measured of the different treatments also imply that flowers that are exposed to honeybees essentially entirely self-fertilize, whereas flowers exposed to native insects obtain fitness that's not statistically distinguishable from cross-fertilization. And these fitness differences are huge. I mean, like not 2 to 5 times, what does that mean? Well, one thing it means for the field of pollination biology is we usually judge the importance of a pollinator to a particular plant by some combination of how frequently it visits that plant and how much pollen is deposited by a single visit from that particular pollinator. So that's the classic way of doing importance, but it assumes that the quality of the pollen delivered is the same. And the differences in the amount of pollen delivered by different pollinators is often, 2-fold, 3-fold, 5-fold, but I'm showing you differences after that in fitness that go perhaps in the other direction. So we really need to incorporate these kinds of things when we're asking how important is a particular pollinator species for a particular plant species. The second part, which is a lot harder to answer, and I'll come back to it at the end and we can speculate, what does this mean for our our flora, that 84% of all visitors are honeybees, and yet honeybees provide pollination services that lead to less fit offspring. So I'm going to leave that because ecology is difficult, and making prediction there, I can give you some scenarios that and I will, but I won't answer that immediately. Instead, I want to say one more thing, is that we scoured the literature and found 50 plant-pollinator interactions. Some of these are crop plants, some of these aren't, to ask whether honeybees consistently visit more flowers per plant than do other insects that visit the same plants. And the answer is a little complicated, but the more dominant honeybees are, the greater the difference between the number of flowers visited by a honeybee and the number of flowers visited by any of the other pollinators. So it's pretty general and applies both to crops and to wild species. All right, second half, what does this mean for bees? These native bees, largely solitary, not entirely. We've got bumblebees, but I'm arguing that honeybees particularly visit the most abundant pollen sources, and that pollen is food for all bees. So maybe we can estimate how many native bees might be missing because honeybees are taking the pollen. And we do this in two ways. One is my favorite way, because it's the simplest. We convert the biomass of honeybees to biomass of native bees and say, well, honeybees are made of pollen and nectar, and so are native bees. So if that pollen and nectar wasn't used for honeybees, how many more native bees would we have? And the other is that we can calculate how much pollen the average native bee needs to be produced. I'll show you how that's done. And we can ask, given the amount of pollen that honeybees remove, because we're going to measure that, how many extra bees, native bees of average size would we expect? So we need to know the mass of a honeybee and the mass of the average native bee. That's not that complicated, but we need to know how much pollen you need to provision the average native bee, and I'll show you how we do that. And we need to know how much pollen do honeybees remove that could otherwise be converted into native bees. So we stand on the backs of many other biologists who figure out relationships between things. So on the left, we've got the relationship between the mass of a bee and the intertegular distance. That's the distance between the two wings. So there's a nice relationship. If you can measure that distance, you know the mass of the bee. And if you know the mass of the bee, on the right there, you know the amount of pollen it takes to provision a brood cell of a bee of that size. And this is all stuff done in, you scour the literature to find these, what are called allometric relationships. So there's the intertegular distance on that big bee there. It's the distance between the wings. So in order to get the average mass of, or the mass of the average native bee, well, that guy James Hung went around and identified all of the species that were visiting flowers in western San Diego County and had data on their frequency, and he also pinned them all so we could measure their intertegular distances. And so we know then the mass of each species, we know the frequency of each species, and therefore we know how much total biomass of each species there is. And we can, when we know how many native bees there are in the collection, we can get to the average mass of a native bee. Average mass of a honeybee is easy. You just put it, you dry it out and weigh it. Plus, it's all over in the literature. So average honeybee, it turns out, weighs 32 milligrams. The average native bee is much smaller. There's an error here. Laserglossum is actually a little bit smaller than 5.86, but the average mass of a native bee is 5.86. There's a lot of pain involved in getting these. So here it gets real simple. This is my simple-minded way of doing it. 84% of all visitors are honeybees. 16% therefore are native insects. The vast majority of those are bees. That's the ratio of their weights. So you can just multiply it out. And the calculation there is, in the absence of honeybees, if the pollen and nectar used to make honeybees was instead used to make biomass of native bees, we'd have 30 times more native bees than we observe now. The other way has to do with measuring pollen removal and how much pollen it takes to make the average native bee. And I don't want to, there's a lot of calculations. I don't want to belabor this, But we've got the mass of the average bee, and therefore we've got the amount of pollen it takes to provision the average bee out of one of those relationships. We also have to know the percent of a nest provision that is actually pollen, which we can get from the literature. And therefore we can get to the mass of pollen that's required to make a single individual of the average sized native bee. Then we ask how much pollen is being produced by doing a similar thing with measuring pollen grains and their masses and the frequencies of those plants. And also we need to know how much pollen is actually being removed by the honeybees. So I'm going to move on to that part. And I'm just going to concentrate on this slide here. So here again, you go out and you put those mesh bags over inflorescences and come back when the flowers start opening. Take the mesh bags off and allow for, well, there's several treatments. Either you leave the mesh bag on, that's the pollen exclusion treatment. The begin treatment on the left, that's how much pollen is in the anthers when they haven't been visited at all. You measure how much pollen is there at the end of the day. If they stayed in the bag, it's a little bit less because the thing shakes around a little bit, but mostly it's all there if pollinators haven't visited. The next two bars that are highlighted there are first and second visits to the flower by a honeybee and how much pollen remains in the flower, so how much pollen was removed by honeybees. And the answer there is almost 40% of the pollen available in the flower is taken by the first honeybee that visits. And after 2 visits, you've lost 60 to 75% of all available pollen. And if you allow that thing to be visited all day, you end up losing 80% of the available pollen. So that's the amount of pollen that we're estimating honeybees gather. 80% of the pollen from each flower. We then have measured the number of grains and their sizes and all sorts of things. And we can convert that to the number of native bees of average size that could be produced in a hectare that has the density of that flower that we observe while it's blooming each day. So for black sage, Salvia mellifera, the patches that we're looking at are producing about 30 grams of pollen a day. That's how much is in a flower, how many flowers on a plant, how many plants in the patch, all that kind of stuff. And that's enough to make 7,000 native bees in one day. For Salvia apiana, those patches weren't as densely flowered, and they made about 12 grams of pollen a day, which would make about 3,000 native bees. And for Phacelia distans, which is not as abundant a plant, they make about 1.8 grams of pollen each day per hectare, and that's 523 native bees could be provisioned with, of average size, could be provisioned with that pollen each day. Well, these plants don't bloom only one day, so we actually went to iNaturalist and got blooming pictures, and they show nice little curves of the blooming season, and we just took the middle 50%, so the abundantly blooming period, which is about a month from these plants, totally reasonable. And if you then multiply the previous, the figures in the previous slide by 60 days or 43 days, for white sage, Salvia apiana, you'd expect that a patch of white sage at the density that we're observing would make you nearly 200,000 native bees. And a patch of black sage would make you almost 500,000. And a patch of Phacelia, 22,000. I was just on a Zoom call this morning. We're trying to figure out how many native bees are actually out there by various observations we've made. It's not an easy thing to estimate, but probably the number is in hundreds at most. Yeah. So either way we calculate it, in the absence of honeybees, we'd expect far more native bees than we're seeing. So by method 1, we calculated a 30-fold difference in the number of native bees that we might see in the absence of honeybees. And I don't have a similar fold increase for the second way of doing it based on the amount of pollen that honeybees take, but I think it'll come out at least as big of a fold difference between observed and expected. So there's been sort of two parts to this talk. One is what are the effects on plants? And I showed you that pollination by honeybees is the norm here, but it leads to low fitness offspring. So this gets back to that question, who cares and what would it mean? And this is where we get to community ecology, and I'm not a community ecologist, and I'm not a community ecologist for a reason, which is it's really complicated. So I can invent some scenarios. Lower fitness might lead to lower plant cover, and around here, where you have lower cover, you have more grass. And if you have more grass, you have more fire. So that's one way of looking at it. Here's another way of looking at it. The abundant things have low fitness offspring. So that might reduce their abundance and actually help the rare things. So that could help preserve diversity. It's not really where I want to end. I'm just saying lots of things are possible. One thing I will say is I don't think no effect is the answer. It's really hard to know where this leads. But I would say that honeybees have been given pretty much a free ride as invasive species, and maybe it's time to start considering their effects, especially in places where feral honeybees are as common as they are here. The second part, this huge potential for a huge effect on the population sizes of our native pollinators. Again, it's very speculative, particularly because we don't really know what limits the population sizes of native insects, especially native bees. It could be food, but it might be the weather or nesting sites or something else. It's a contentious issue in the entomological literature. I think the easiest thing to say is that it would be very difficult to believe that none of them were limited by pollen, or that this huge amount of pollen that is consumed by honeybees, this huge fraction of all pollen that is consumed by honeybees, has no effect on the population sizes of our native insects. The problem in ecology, and it's a common one, is we don't have baseline data from before honeybees. I would love to be able to remove honeybees from a 20 square mile area and come back a decade later and count the native bees. Probably nobody's going to let me do that. And I won't live long enough, and actually doing it is very hard. So it's hard to know what we can actually do about this situation. But the effects are potentially quite big. All right, I'll leave you there with some bee faces. One of these is a fly. So people that know bees make these things, which isn't me, and they always put a fly in or some other insect. And yeah, so I always fail on this. So I'm glad you know. So let's see. Somebody asked about pollination services by honeybees. You've all heard that there's some sort of honeybee apocalypse. And the facts of that are that, in fact, just 2 weeks ago, the USDA has the Department of Statistics put out a study that said we currently have more managed hives of honeybees than we have ever had in the United States. The problem honeybees do face is overwintering survival has been reduced. They have a number of diseases and effects from pesticides and many other things, such that the work of a beekeeper is much increased. So in the spring, they have to split their hives much more frequently than they used to in order to get those stocks back up. But we're not running out of honeybees, at least so far. So let's see, any other questions? Yeah.
Actually, two questions. At the beginning of your talk, you said something about some plants that don't allow self-pollination.
Yeah, so about half of all species, and that would include almonds and apples, cannot be fertilized by their own pollen. So that's called self-incompatibility. And I spent 20 years studying that, so you can ask me anything.
So would you say then that native bees are thriving on those species because they kind of have
Well, the numbers of native bees on those species as grown currently is pretty low because the habitat around the agriculture has been blasted by and large. So this especially applies to sort of the southern Central Valley for, say, almonds. There you'll get in the upper 90% of all visits from honeybees. What is noticed in careful studies is that a single visit from a honeybee, even though it deposits a reasonable amount of pollen, doesn't result in as much fruit set as a visit from a native bee, probably for the same reason. The honeybees are mostly visiting. In fact, it's hard to understand why you'd ever leave a tree. You know, you've seen an apple tree in bloom. It's got a zillion flowers. Why would you ever go to the next one? And honeybees generally don't. So they work well enough, and they can be managed and imported in huge numbers. These native bees that are solitary, that you just can't do any of that. So honeybees are going to maintain their position as the workhorse for pollination. But if you actually start studying the effects of the few other things that visit, often they're quite important. And there's one study that came out in Science some years ago that said no matter how many honeybees you have, the number of non-honeybee visitors still increases seed or fruit set in the crop. And this is across many, many crops across the world. So I would say that the people that work on this are recognizing more and more that non-honeybee insects are really important, even in crop pollination. So there's moves afoot to grow vegetation on the margins of agricultural land that will allow these native bees to persist. I mean, the thing about native bees, they have pretty short seasons individually, but they're not going to coincide exactly with the flowering phenology of the crop. So if you get up in the southern Central Valley where the almonds have taken over, you know, we grow 80% of the world's almonds now, and there's not a lot of vegetation that's not almonds. And so there's not a lot of native insects, but there are some management practices now that are recommended or being studied that involve actually messier farms. Yeah.
Did I understand you correctly that the feral honeybee population is much greater than the hive honeybee population?
That's my feeling. The reason I got started on that was that when we collected bees to do the genetic analysis, we just went around. We didn't go to hives. We didn't go to feral hives, or we just went around and grabbed workers off of plants right across the county. And every single one was this genetic mixture. And we did this back in the time when the recommendations were to try and maintain ethnic purity, to try and because the scutellata hybrids are known to be more defensive, harder to. There were efforts to try to keep them out of agriculture, and so if those were common in the system, then we would have seen some purely European bees, and we never, every single one 40% African ancestry. Plus, if you go out to any of the mesas around here that have a stony outcrop and start looking in the holes, you can find honeybee hives. So these honeybees, these scutellata hybrids, tend not to nest so much in tree holes, which is a good thing because we don't have a lot of trees, but mostly in the ground, either in boulder fields or pockmarked mesas that have cavities in them. The other place they like a lot is irrigation boxes, those things that have the valves in them. So I often ride a bike on the 56 bike path, and you don't see it anymore, but there was a time when every single one of those, you've got that little hole in the top that you put the tool in, you can raise up the top and fix the valve. Every single one had a kind of a hard foam in it because the county didn't want people to open those up and suddenly be swarmed by scutellata hybrid honeybees. Now most of those have fallen out. It's not clear how much more defensive, they used to say aggressive, how much more defensive these Scutellata hybrid bees are. We've actually tried to measure that. It's super hard to measure. But there are accounts, of pretty serious attacks occasionally.
A related question, do the diseases that affect the hive honeybees spread to the feral population?
Yeah, so I was very interested in studying this, although I had no background in it. So we do have one honeybee biologist on staff at UCSD, and one of his students at my behest started to look at viral loads in feral and managed hives, and they're roughly equal. So these feral bees do get the virus, but it doesn't wipe them out. So they're either they are likely to be more resistant for any number of reasons. Could be genetics. They also do things like they swarm a lot. So now it's springtime. You may have read that the Dodger game was delayed just yesterday by a swarm of honeybees behind home plate. It's a great story. They called up a bee remover. He was 45 minutes away and rushed in. It took him almost 2 hours before the game started, and they had the guy in his bee suit throw out the first ball. So they move home a lot, and they move home more than, and when they do that, there's a break in the larval cycle, and it's the larvae that the Varroa mite, which is the vector for many of these diseases, feeds on. So it could be that they're more resistant for that reason too. That would not be a great trait for managed hives, but it might work for the feral ones. So we don't know. There's a whole lot of available avenues of research here. But not being a real honeybee person, I never really went down that road other than to say, hey, look, we've got these bees that are super successful. They don't get any help from humans. Maybe they would be useful. And these days, beekeepers actually are starting to keep these because they think that they survive better, even in hives, than do the purely European forms of bees that they were working with up until now. So it's not clear what's going to happen policy-wise or anything about whether, for a while in San Diego, it was policy to requeen your hive every 2 years with a queen who was inseminated outside of the range of quote-unquote Africanized honeybees. But I think that beekeepers have taken the law into their own hands now, and they actually think these feral bees make good stock, despite their occasional ones that get aggressive.
Thank you very much.
Yeah, thanks for listening.
I just wanted to thank you guys again for coming to the final talk tonight. We will have new topics and new speakers come 2025, February. Gabrielle and Karen are amazing at choosing topics and speakers. So really look forward to seeing you guys next season. And thank you so much again for coming. Drive safely.
Thank you.